and welcome to welcome to Bioclues Bioinformatics School Children Cohort Week 4, 2022, and I welcome all the children. And today we have a lecture on microbiology and genetics by Aparna Banerjee, Dr. Aparna Banerjee. So Dr. Aparna Banerjee, she has done her PhD in botany from the University of Burdwan, India. She has bachelor's and master's in biotechnology with her master's specialization at pharmaceutical and medical biotechnology. So she has done her postdoctorate from University Cattolica del Mole, Chile, where she is presently working as an assistant professor. In her last 8 years of research experience, she worked on extremophilic microbiology. Her interest area includes that biodiversity, conservation, and bioinformatics. She is presently a core member of India's Bioinformatics Society, Bioclues organization, and her achievements include like she is running 4 international projects as a principal investigator and co-investigators, and more than 30 publications in nationally and internationally reputed journals. So she has featured in the documentary podcast in UK, featured in the World Campaign 1 Million Women in STEM in 2020, also featured in UNICEF India programs on climate change. She got awarded as a Young Scientist Award in 2018 in Indian Science Congress Association. And so also she has awarded Swarna Jayanti Puraskar in 2017 from National Academy of Sciences India, and she has been selected as a Young Researcher Forum at the 10th International Conference on Nanomedicine and Nanotechnology in Healthcare 2016 in Bangkok, Thailand. So here is the info, and I welcome Aparna. Here goes. Your voice is not audible.
Okay, sorry.
Yeah.
Well, I was muted. Thank you, Shantoshi, for the introduction. So, should we start now?
Yeah.
Okay. Is this visible?
It is visible.
Okay. So, I think before I start, I would love to hear from the students who are present over here if they want to talk something, or they want to at least let me know their name and who they are so that we can start.
Absolutely, Dr. Banerjee. First of all, a very warm welcome to the stage, and thank you so much for being here today. It's genuinely an honor to hear you, and I'm really looking forward to your talk. My name's Naya Mehta, and I'm currently in the 12th grade. I'm very interested in, microbiology, genetic engineering, and also the mathematics behind biology. And so bioinformatics was something that I found myself drawn towards. Yes, and like I said, I'm really looking forward to your talk. Thank you for being here again.
Thank you, Naya. Anyone more?
Hello, I am Aradhya and I study in 10th standard.
Okay, Aradhya. Nice to meet you. And anyone more here other than Naya and Aradhya?
I think Gunjan Advani.
Okay, but through the classes we will speak, and please feel free to ask at any point because this is more than a class, like a discussion. So if you have any queries at any point of time, just make your microphone on and speak it. So at that point only we can resolve it, okay?
Yes, ma'am.
Okay. So today we are, I think before in the last 2 days, you have already learned a little about bioinformatics, different parts of bioinformatics. So today we will discuss a little about bioinformatics in microbiology. You have probably learned something related to the macro system. Today we are going to learn bioinformatics, but on microsystems.
Aparna, 1 minute, Can you just make it a full screen?
It's not full screen? Okay.
Yeah.
Okay. Um, I kept it. Is it full screen now?
One minute. Still, it is not.
No. For me, it's actually showing full screen. I don't know what's going on. What should I do?
Can you go for slideshow?
Yes.
Yeah. From the beginning, you can just.
Is it fine now?
Yeah, but still it is the same.
I'm making full screen actually. I don't know why it's not showing there, the full screen. What should I do?
Aparna, is the PPT is in the website and the desktop?
It's in desktop, yes.
Okay. I think just need to refresh and then do it again.
Please rejoin, please refresh and rejoin and share the full screen. That will work sometimes. That happens.
I'm just doing the entire screen. I think that will be easier.
Yeah, entire screen.
No, you could show the refresh. Yeah, no, it works.
Yeah.
It's fine?
Yeah.
Okay. Sorry. Okay, so before we start bioinformatics in microbiology, I will talk a little about what is bioinformatics and how it started. So basically, in 2020, 2 years back just, we had 50 years of bioinformatics, because the term bioinformatics was coined in the beginning of 1970s by two Dutch scientists, Pauline Hodgweg and Ben Hesper. And actually, Pauline is still now working in bioinformatics. She's a Dutch scientist who works on theoretical biology and informatic systems of the cells. So that's the time that this term bioinformatics started. And they basically describe they coined the term bioinformatica in Dutch language, and from there this term bioinformatic came. So it was like, the way they described it was the study of informatic processes in biotic system. So biotic system include all plants, animal, microbes, all the living beings that we have are biotic system. But as the time passed on from 1970s to 80s, for any concept, the concept we renovate it with the time. So one of the recent idea about bioinformatics in 2001 was, bioinformatics is the application of computational tools to organize, to analyze, to visualize, and store the information associated with biological macromolecules. So when the term macromolecules come, macro is like big, and molecules we know that are small compounds. So macromolecules is bigger size of molecules. For example, a protein is a macromolecule, a carbohydrate is a macromolecule. So what is the molecules over there? The proteins that are formed, that are molecules there, and when a lot of molecules are together, they form macromolecules. So the system information related to DNA, related to RNA, related to protein, related to carbohydrate, all these composed of bioinformatics. So when we think about bioinformatics, also there a term comes, that is computational biology. So we sometimes think that bioinformatics and computational biology are kind of similar. There is a thin line of difference that computational biology deals with the modeling of biological system, whereas bioinformatics deals both. It develops the software or tools and the algorithms, and then analyze and interpret the biological data of macromolecules using a variety of software tools and algorithms. So both composed of bioinformatics. Now, as today we will think about bioinformatics in microbiology. So we should think, what is microbes? When we talk about microbes, we normally think about bacteria, virus, fungi, because these are the things that we are normally accustomed to hear. Probably nowadays also virus for COVID-19. So the term virus is very common to us. We are listening it every day. Bacteria, obviously, we listen, and then fungi, we listen for infection, for foods. But apart from that, there is also, for microbes, there is no prokaryote and eukaryote. Anything that we cannot see by our naked eyes are microbes. So can be bacteria, can be archaea. What is the difference between, till a time, archaea and bacteria used to be confused and given the term archaebacteria. But then archaea and bacteria are now two separate groups, and archaea are normally found in extreme ecosystem. For example, our planet Earth is composed of a lot of hot springs, a lot of cold ecosystem like Antarctic and Arctic ecosystem, like the Himalayan glaciers, in the deserts, or under the sea, in totally deep sea where pressure is high, there is hydrothermal vents. So all these extreme ecosystem, archaea can be found. In the limit of life, they can be found. Fungi can be also found everywhere. Algae. So algae can be macroalgae and can be microalgae too. So microalgae are also microbes, and microalgae are normally found in water bodies. Probably many of us can see when we are near a pond and we see the pond from the far is totally green, or the pond from the far is totally yellowish. So that basically a bed of microalgae formed over there. And protozoa also. And we also know probably of a lot of protozoal infection that cause due to food poisoning, lot of amoeba infections. So out of this, bacteria, archaea comes under prokaryote, fungi, algae, protozoa, they comes under eukaryote. Now bacteria and archaea, they are unicellular, but when we are talking about eukaryotes, they are multicellular. But even being multicellular, we can see them. We cannot see them with our naked eye, and we need to see them with the help of a microscopy. So they are called microbes. And virus, that neither comes under the entity of prokaryotes or eukaryotes. So the term microbes is a bigger term. It's not a classification. It's composed of a lot of living beings and viruses that we have in our planet Earth. And we can find it everywhere, in water, in air, in soil, everywhere. Wherever we are seeing life in planet Earth, or in our naked eye, we cannot see life. For us, it's a extreme limit of life. Microbes are there. So today we are going to focus more on bacteria. And if we talk about bacteria, so bacteria are mostly found in for example, we are in a room and the room is empty. The room only have air, and microbes are there in that air. So microbes can even spread through the air via their spores that they form. Microbes are present in soil. So for example, we can even see which kind of plants in their root have microbes. If some of you have seen pea plants, probably if we have a big garden, all of us must have at one point of time pea plants. So if we take out the pea plant from the soil and we see the roots, we will see that the pea plant roots have some nodules. And inside that nodules, microorganism lives. So what they do, they take the nutrients from the plant and instead they give the plants nitrogen. So they basically help plants to live. They basically help plants for their living being by giving them nitrogen, supplying them nitrogen. Otherwise, we need to supply nitrogen to the plant via fertilizers. So there comes the term biofertilizer, because the bacteria that are living in root rhizosphere or the soil, the vicinity of the soil for the plant, um, they help the plant. So there is a symbiosis between both of them. And also the bacteria or bacteria is there in water bodies, in all the water bodies, in the stagnant water bodies, in the coastal ecosystem, in marine ecosystem, in freshwater ecosystem, in deep sea, everywhere there is bacteria. So when we think about bacteria, there is a term that we always think that microbes are dangerous, microbes are bad. So there is a concept good and bad bacteria. Actually, we talk very little about good bacteria, but there is probably a thing that bacteria, that the good bacteria, the term, the concept is bigger compared to the bad bacteria. Even the bacteria that is living in our planet earth and not exactly within our microbiota, within the human body or within the animal body or within the plant body, only in the environment, what they do, they take part in biogeochemical cycles. And why biogeochemical cycles are important? So, for example, we have carbon, we have sulfur, we have nitrogen, we have different elements in our planet Earth. And what the bacteria do, they basically cycle them. So, for that, all these compounds are getting cycled and recycled. And if at one chain the microorganism or the bacteria stopped working on that particular biogeochemical cycle, so what happens if the sulfur cycle is not working, if the carbon cycle is not working, if the nitrogen cycle is not working, that entire ecosystem get hampered. And when an entire ecosystem will be hampered, all the ecological niche in the ecosystem where the animals, the plants, all the living being lives, their food and their food chain will be blocked. So in a way, they are not directly interacting with us, but they are doing something good for the planet Earth, for the ecosystem. Or many bacteria also do photosynthesis. So in a way, they are producing oxygen. Actually, a major quantity of oxygen getting produced by the marine photosynthetic organisms. So in the entire marine ecosystem that we have of the world, the majority are marine ecosystem. So there they are doing the photosynthesis and we are getting the benefit of them. Even if we talk about microbiome, where the bacteria lives within the body, any particular body, for example, the example of plant that we were talking, that the microorganism lives within plant root, like nodules, small nodules, and they help the plants by supplying them with nitrogen and instead taking nutrients from them. Plant microbiome. So in all these microbiome, they are helping in some way. Like maybe we all talk about this nowadays. Nowadays, we all are actually aware of functional food. So even in India nowadays, we see probiotic drinks like Yakult. I see in Big Bazaar. Probably many of you have seen. So what this Yakult do? There are many other marks also other than Yakult. If you see the composition of Yakult, have a different composition of lactobacilli. So, for example, when our stomach is upset or for some reason our intestine, for any immunological suppression, our general intestinal flora is getting down, we take this small pack of Yakult, we drink it. It's somehow like a drink of curd, but it's not exactly curd. It's a small drink with concentrated bacteria. And the bacteria does what? When they are inside our intestine, they again colonize and they help us to establish by establishing good microbial flora inside our intestine and helps us in digestion also. So what are the common good bacteria that lives lactobacilli, bifidobacterium, Escherichia coli? We have all heard of these bacteria. So they are helping in maintaining our own body health. And interestingly, the number of bacteria inside our body, number of microbial cells inside our body are way number of the animal cells that we have. And what are the example of bad bacteria? actually sometimes, for example, in skin, we have staphylococcus. And this staphylococcus don't remain as obligatory pathogen in our skin. They remain in our skin. And when, for example, we are immunologically depressed, we are immunologically suppressed, we get up from some disease, we had some prior skin infection, we were getting treated with some medicine that took the entire good microbial flora from our skin. So the staphylococcus makes skin disease at that time. So there are bad bacteria also living in our body, but they are during immunodepressed condition, they are uh, becoming to be a pathogen. Also, there are pathogen in the environment that we are getting infected regularly. Can be airborne pathogen, soil-borne pathogen, waterborne pathogen, and so that's a part. But a majority part of the microorganism or bacteria in our daily life, we are using them and we are getting benefited from them. And the example that I was talking to you, that even where we think that there is no life, where there is life, even in that kind of extreme limits of life, the bacteria lives. So if some of you have went to Himalaya and you have seen snow in that in that permafrost region or polar region or sea ice, bacteria lives, archaea lives. In mud volcano is there in India also. If some of you have went to Andaman or knows about Andaman, there is Baratang mud volcano in Andaman. So in this kind of mud volcanoes, bacteria lives. They lives in hydrothermal vents, in fumaroles. Fumaroles is not exactly hot spring. Fumaroles are from where due to geological activities, lot of gases, acids, and mixed of soil and water is coming out. Fumaroles, mud volcanoes, hot springs, hyperacidic lakes, in desert, like in India we have Thor desert, in acid mine drainage, in the anoxic region of deep sea, the deep sea hydrothermal vents, even in nuclear contaminated sites, in all this kind of ecosystem, which is extreme, can be man-made, like the nuclear contaminated site, or can be the original ecosystem of planet Earth, bacteria lives. In Gujarat, there is this the area of Kutch where there is soda lake, hypersaline lakes. So even in that kind of salty ecosystem, the bacteria lives, the archaea lives. So they are living, they are helping in that particular biogeochemical cycles of those extreme environment. And even in this time of climate change, because they are living in those extremes, they are maintaining those ecosystem. And if during climate change we lose those bacteria, basically we will lose slowly those ecosystems too, because there will be no other microorganism or no other organism to cycle the elements of that particular ecosystem. So in a way, they are also helping in balancing the life of planet Earth. So for all this, we never see bacteria, isn't it? That's the problem. We talk about bacteria, we are aware of bacteria, that they are doing good or bad, but we cannot see them. But we can see with a microscope, but really we look them very small and we cannot see them in a bigger way, isn't it? Probably in school you have seen a compound microscope where there is an optical microscope with some lens system. The lens can be either with an external light source like sunlight, or there can be a bulb and you put it with a connection with electricity, and then there is a bulb light and you can see the bacteria. So what we see bacteria probably in 60 magnification or 100 magnifications, very small, can be some small dots, can be a rod, can be a chain, can be some comma shaped things. And in 100 magnification, we put some oil and we see them a little more bigger. I don't know if some of you have in school have seen microbes or from water or from plant cell or not. But you can do a very simple experiment if there is a compound microscope in your school, or even nowadays, there are kits of microscope available in the market. So if you have interest, probably you can buy those small microscope kits, and you can just put a water drop. You can just put one small piece of a of any plant, and you can see them if there is, you can see something moving or not. So to identify the bacteria and also to classify the bacteria initially, gram staining is one of the method that is widely used till date. There are few bacteria like Mycobacterium cannot be stained with gram staining because they have mycolic acid, that is lycoarcs, and the stain don't pass through the bacterial cell. But this gram staining, generally they are widely used for classifying the microorganism. So what basically gram staining do, we have a series of stain, crystal violet, and then we give grams iodine, then we wash them with a series of alcohol, and then we put saffronin. So all these stains do what? If the crystal violet pass inside the bacteria, and with grams iodine or mordant, we fix the bacteria with crystal violet. The term violet means what? You can see even in this picture that some bacteria are violet colored bacteria here. So these violet colored bacilli or rod shaped bacteria are gram positive bacteria because crystal violet entered there and the bacteria got stained, and you can see it's like a violet. What is saffronin? Gram negative bacteria. The gram negative, the positive point of gram positive bacteria, why they get this violet stain is they have a bigger cell wall. So for this cell wall that they have, when the stain is getting entered inside the cell wall and you wash them with alcohol, the stain don't get out. But in case of gram negative bacteria, the cell wall is really smaller, really thinner. So when you wash it with alcohol, you can wash the crystal violet. So when you put saffronin, you can see saffronin is pink in color. You can see pink gram negative bacilli or rod shaped bacteria. So that way, due to the cellular composition of the bacteria, you can see them as gram positive or gram negative. So the gram positive will always look as violet and the gram negative will always look as pink. But how we can see, okay, so who developed this technique? It was a Danish bacteriologist, Hans Christian Gram, who developed this technique long back. But with the time, definitely, we had more inventions in microbiology. So there is scanning electron microscopy, there is transmission electron microscopy, there is confocal microscopy, atomic force microscopy. All have different functions. In electron microscopy, we see the cell really bigger. So for scanning electron microscopy, we can see the surface of the bacteria. For example, if the bacteria is producing some compound, if the bacteria is getting attached to some another bacteria, having some cellular communication, talking inside a colony, cellular communication. So we can see more or less the surface of a bacteria, the outside, the surrounding of the bacteria. Whereas in case of transmission electron microscopy, we can see inside them. What is inside the bacterial cell, we can see with transmission electron microscopy. What we can do with a confocal is totally different. With confocal, we can put different stain and we can see separately the gene, the cell wall, the cytoplasm. So with different kind of stain, we can see different parts of microorganism. And with atomic force microscopy, we can see the width of the bacteria. With none of this microscopy, we can see the width, but with atomic force microscopy, we can see the width of the bacteria. So there are different other type of microscopy also. If you go to maybe you can go to the Nobel Prize website and you can see that in different years, there have been Nobel Prize for invention of different microscopy. So the inventions are going on, and probably someday we will have another different kind of microscopy where we can see the microbial cells more bigger than we see nowadays. So there is a way that we can see the bacteria by microscopy. But even if we see the bacteria via microscopy, how to identify them? There where comes bioinformatics. Bioinformatics basically help us. Bioinformatics is the torchbearer to identify a bacterium, because we can say one bacilli. Why a bacilli? Because of the rod shaped. We can say some staphylococcus or some cocci. Why cocci? Because it's round in shape. But we cannot distinguish more than that. We can only say that this is bacillus or coccus or comma-shaped bacteria, nothing more than that. We cannot exactly identify which particular bacteria is that, but we need to identify them, isn't it? Otherwise, how we will know that for which function those bacteria are doing? There comes bioinformatics. So for microorganism, basically, or bacteria, we do 16S rRNA, 16S rRNA gene sequencing. What is 16S rRNA? The ribosome that the prokaryotic ribosome have is 70S ribosome, isn't it? 70 is Vedbarog unit. Probably you all have studied this. And in 70S, there is 50S and 30S. So this 50S and 30S, this large subunit and the small subunit together is a 70S one. So this large subunit 50S is composed of 23S, 5S and some ribosomal proteins. And the 30S, the smaller subunit, have 16S and ribosomal protein. So this 16S basically is universal in the microorganisms. They are basically conserved in the microorganism, conserved sequence, and they are widely present in all the bacterial species. So when we are trying to identify someone, maybe all of you have done plant identification in the school practicals, isn't it? So for identifying plant, there are different techniques, that the leaf should be like this, the stem should be like this, the root will look a little different. So we put a category common for a large group of plants, and that's the way we can say, okay, this is a rice plant, okay, this is a wheat plant. In case of microorganism, because this 16S is widely present in all bacterial species, and these are also housekeeping genes present in all the bacteria, so this 16S gene sequencing is one of the most common methods for bacterial phylogeny or genus or species classification. With the time, it's really being difficult to species classification, because often the species are really very similar. For example, there is a bacteria Bacillus cereus and there is bacteria Bacillus anthracis, there is bacteria Bacillus thuringiensis. All are different bacteria, but they are so near that it's really difficult to classify them exactly till species by 16S rRNA gene sequencing. But yes, we can say that they are any of three of them. So what it do, 16S rRNA gene sequencing, they help differentiating very closely related bacterial species. For example, they can differentiate between a Bacillus subtilis and a Bacillus cereus. Maybe Bacillus subtilis and Bacillus cereus looks really similar in the microscope, but when you do 16S gene sequencing, we know which is the which bacteria. So even in the clinical laboratories that we have nowadays in India or in all over the world, the laboratories rely on this method. They do 16S gene sequencing to identify the unknown pathogenic strain. For example, many times we see in the television news, in the channels, an unidentified disease have came, an unidentified pathogen have came. So how to identify those pathogen? The clinical laboratory rely on 16S gene sequencing. They isolate that microorganism, isolate their DNA, do the 16S gene sequencing, and came to know which bacteria was the pathogen. So according to and when we are talking about classification, there should be the proposed guidelines also. So what is the guideline for bacterial classification? That the strains with less than 97% similarity in 16S rRNA gene present different bacterial species. So if it's less than 97, they are definitely, so when you are doing the sequencing and then you are trying to find out their similarity, if they are similar less than 97%, they are different bacterial species. And when it's more than 97% similarity, you should classify it with alternative approach. So why we should do the 16S gene sequencing? To give us really easy example to understand that microbes are there in our daily life. We are using microbes in medicine. We are using bacteria in medicine. in nutraceuticals, like when in an immune suppressed condition we are taking the lactobacilli. We are using the bacteria in the rhizospheric microbiome for the development of plant growth. What they are doing, they are helping the plant for growth and development. They are helping the plant to uptake nitrogen, phosphorus, potassium, nutrient acquisition. They are helping the plant in their physiology and metabolism. They are helping the plant to fight with pathogens and also to tolerate different stress like temperature, salinity, water stress during drought conditions. Also, we are using bacteria nowadays in wastewater treatment plants. So, for example, in a city, every day there is some water waste. A big amount of water is getting thrown out to a bigger water bodies or to a water treatment plant. In the water treatment plant, we are using microbes to treat the or to flocculate the waste inside that water. So we are using microorganism or bacteria even to treat wastewater, because microbes or bacteria work as a flocculant. What the flocculant do? That you use the flocculant in a water with different things, the flocculant bind with those particulates and make them precipitate. So in turn, what we get in the wastewater treatment plant, clean water in the surrounding, in the up, and all the, not exactly all, depending on their size also, but the majority of the pollutants get precipitated down. Also, we eat them in our regular life. Maybe in parts of India we eat dosa, we eat idli, we eat all the fermented foods that are with microorganism. We all eat paneer, that is also there is the presence of microorganism. We eat different kind of cheese where there is presence of microorganism. Curd, there is presence of microorganism. All kind of, there is sauerkraut that normally we don't eat in India, but outside India we eat it. There are different kind of fermented drinks. So all these fermented products are basically made with good bacteria that helps us to stabilize our own intestinal microbiome and helps our body to uptake nutrients for the growth and development of our body, for nutrient acquisition, and many more. So every day in our life we are using microorganisms or bacteria in different purposes, and that is the reason we need to know them also, that what is their role in our body or who are they. So during 16S gene sequencing, what we do? We isolate the bacteria, we take out the DNA, we extract the bacterial DNA, we amplify the 16S rRNA gene. How to amplify them? There are primers. So when we know that these are universal 16S rRNA gene, the primers will be these. The primers are available in the market, and with a machine called thermal cycler machine, PCR reaction, polymerase chain reaction. Probably you all have heard also about this during this type of COVID, PCR, RT-PCR, PCR. So this PCR, polymerase chain reaction, done in a thermal cycler machine, amplify the 16S rRNA gene and we sequence a portion of the 16S rRNA gene, and finally what we do? We compare the sequence in the database. So there is a database, gene bank, or there are many other. We put our data in that database. They search all over the database where they get a match, and when they get a match, they let you know, okay, so this unidentified DNA is this bacteria. So that's the way this technique basically function. So probably now we can try a little with finding, with doing a little work in the computer. Are you all having your computer with you?
Yes, ma'am.
Okay. Let me. Extremely cold it is. I feel little suffocation. Extreme cold it is, and even in the cold, sometimes the AC stopped functioning for deep freezing, so feels more cold. Is it visible, the screen?
Yeah.
Yes, ma'am.
Okay.
So we will go to NCBI. Okay, probably you have done it in the last two classes also. We will open NCBI. We will do slowly. Okay, we have no hurry, so we will just do slowly. Are you there in NCBI?
Yes, ma'am.
Okay. So here in all databases, we will do genome. We can do genome, we can do nucleotide. When we click on genome, we can basically see the entire genome of the bacteria, the entire genome. And when we do nucleotide, we basically see a part of the genome. So 16S is a part of the genome. If you click on nucleotide, you can search exactly the 16S. If you go for the genome, you will see the entire genome of the bacteria. So when we go to nucleotide and put any bacteria name that you know, any bacteria. For example, I put Escherichia coli. Probably you all have heard this name.
Yes, ma'am.
When we didn't write 16S RNA, you see how many Escherichia coli sequence are there? A lot. It's almost 55 lakhs of sequence that is present in the organism, you can see. So this is Escherichia coli, one particular strain, 16S ribosomal RNA partial sequence. You click it. You can search any bacterial 16S RNA like this. So if you go to this option, run blast, what this run blast do? This option run blast, they basically find regions of similarity between this sequence and other sequences using blast. BLAST is basic local alignment search tool. So what basically BLAST helps, this tool, they basically find the similarity of your bacteria with all the other sequences present in the gene bank, present in the NCBI website gene bank database. So if we go to this option, run BLAST, are you till now there in run BLAST? You are able to click it? Or should I stop?
No, ma'am, there, ma'am.
Yes, ma'am.
Okay. So look, in BLAST, there is option. This is in BLAST N, nucleotide blast. So our option is nucleotide blast because we are taking a nucleotide, isn't it? 16S RNA. BLAST P for proteins. Search a protein database using protein query. So in this, you can do you can search protein sequences in NCBI and do BLAST P. Then comes BLAST X. Search protein database using a translated nucleotide query. When the translation is done, T-BLAST N. Translated nucleotide database using a protein query. You are mixing between nucleotide, between protein, and T-BLAST X. Translated nucleotide against translated nucleotide query. So we are going for BLAST N. So here, in where there is choose search set, there is look the option of database. Here you will we will select the 16S rRNA by rRNA ITS database. So there you can see 16S, 18S, 28S, and the ITS for fungi. So these are basically for all the identification. For 16S for bacteria and archaea for the prokaryote, 18 for fungi, 28 is also fungi, and the ITS region for also fungi. So basically 16 is for the prokaryote and the rest are for the eukaryotes. So for all the classification, using the conserved region for the housekeeping genes, 16S, 18S, 28S, you have to click the option rRNA or ITS database. Then we can, for simply, we don't need to go for the, if there are different other options, if your microorganism, if a uncultured bacteria that you don't know what it is, you don't want to make it paired with the uncultured bacteria, the bacteria that is not known, its identification, you can click on that. So if we don't want to exclude anything, we will not select anything here, we will just go to the option blast. It will take a little time to come. Are you still now there, or you came to the option of blast and you are like in this part?
So ma'am, we are here after
In the,
clicking on blast.
you have put the option, you have clicked the option blast,
Yes ma'am.
and you are in this page, same like the where I am, isn't it?
Yes ma'am.
Okay. So you can see that it's showing you the alignments and they are showing some score, isn't it? So the identity somewhere is 100%. Look, with the Escherichia coli strain, it's showing 100%. Escherichia fergusoni showing 99.24. So then it's showing different kind of identity, percent identity, and you can see the e-values. So probably what we can do, look, there is 100 sequences selected. If we want to make a phylogenetic tree of this E. coli or any other bacteria, we can probably select the top 10 to see the phylogenetic tree in a cleaner way. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we selected 10 sequences, and what we will go in this distance tree of result. If we will click this, will take a little time and the tree will come. You have been able to come till here?
Can you repeat what you just did now?
Yes. Are you in this page?
Yes ma'am.
So you can see probably there was 100 sequences selected, isn't it?
Yes ma'am.
Just deselect that, because 100 sequences comes with very less identity. Go to the down, so we probably don't need them, because what we see that less than 97% are not same. So probably what we can do, we can probably just select to try. You can select as many as you want, but the tree will be bigger. So just for now to try, deselect it and select the top 10 one by one. Have selected?
Yes ma'am.
Then if you go to this option, distance tree of result, look, there are 4 options: gene bank, graphics, distance tree of result, and MSA viewer. Here also in the show you can do 10, 50, 100. So if some of you are facing the problem by selecting one by one, you can click here 10, you can click here 50, you can click here 100, the way you want. Normally the default setting comes with 100, you can change that. So did you click distance tree of result?
Yes ma'am. Done.
Then something like this is coming, is it?
Ma'am, it's loading.
Yes, it will take a little time depending on the speed of internet. Let us wait until the time it comes.
Yes ma'am, it is coming now. It is showing the tree.
Okay. So here, look, here the entire name comes. Here you can play with it. You can, if you put taxonomic name, it will only show the name of the bacteria and nothing more. You can just play with everything here. If you put the sequence ID, so the taxonomic name with sequence ID will come. Look, the sequence ID also with coming. So it's looking a little more cleaner. So the tree method, there is fast minimum evolution and neighbor joining method. These are method of making the phylogenetic tree and has been done long back, years back. So neighbor joining is one of the most used method for phylogenetic tree preparation. There is a little change you can see with neighbor joining. You can also maximize or minimize the differences, but you can keep the difference 0.75, which is a default one. So in the option tools, you can go to the option download and you can download them as PDF file. You can play with the labeling also with here for the format. But if you just go to the option tools and then you go to the option download, you can download it as PDF file. And you can clearly see that this was our query strain that is showing as yellow. And you can see with which it is showing maximum similarity. There are many Escherichia strains in the top 10, Escherichia marmot, Escherichia coli, Escherichia fergusoni, Shigella, Pseudoescherichia, Escherichia. So there are different bacteria that is showing the similarity with it, but the maximum is an Escherichia coli. And that's the way we get the identity. Here we played with a one that was already submitted in NCBI, but we can also submit our own sequencing data in NCBI, in GenBank database. There is an option of uploading your own data. If you are doing some sequencing, you are uploading your sequence, you can upload it and it will do the same way, BLAST, and will give you a phylogenetic tree. And you can with the phylogenetic tree, you will have the idea. And also with this table, look, with this table, you are itself getting this idea in the last stage, the table that we got, we are getting with which 100% similarity is showing. Many times come where it shows 100% similarity with more than 1, 2, 3. So in those cases, what we can do is we can go for other DNA hybridization, and there are other methods for where there is this kind of conflict. In very few cases, we can go for other identification approaches. But this is in general, the universal identification approach via 16S sequencing. When we have the 16S sequence, we go to NCBI GenBank database, we submit them. After submitting them, we do their BLAST, and we can see the tree, the phylogenetic tree. Is it clear till now?
Yes, ma'am.
Yes.
Okay. So I will just show to you to see only that how it looks. So you in nucleotide, you can search any bacteria that you know. Probably we all know the name Lactobacillus. There are a lot of Lactobacillus strains, so probably Lactobacillus acidophilus 16S rRNA. And look, the same way it came. We click, there are a lot of Lactobacillus acidophilus 16S rRNA gene sequence available. We click on it. We go to run BLAST. We select the 16S rRNA and we put the option BLAST. You can put the name of any bacteria in this part and will be the same. So the 100 sequences got selected. We can select 10 sequences. Now we go to the distance tree of result. And in distance tree of result, we can only select the sequence ID because otherwise it will look very bigger, probably the taxonomic name with the sequence ID. And you can see that lactobacillus with all other lactobacillus acidophilus, it's showing similarity. But with lactobacillus gallinarum, lactobacillus helveticus, lactobacillus amylovorus, no. So you can clearly see the differentiation of the classification, how acidophilus is different from the other lactobacilluses. And the same way you can change here the methods also. You can go to the option tools, download, and you can download the PDF file. Okay. So we were playing with nucleotide. If we go for genome, you can see the entire genome of the bacteria. Genome. Let's, for example, search Escherichia coli. And you can see how many genomes are there assembled. 30,324 total genome of Escherichia coli are sequenced. And that's the way their dendrogram is showing all these bacteria that are sequenced from different parts. So we can click on that gene assembly and we can see all the place, all the name, 1, 2, 3, 4, and we can click on the assembly, we can click on the biosample. From there we can download the sequences total and we can do many analysis. So look, there are 598 pages. You can, and there are a lot of total bacterial genome is available. So there is different sequencing method for that. There are sequencer machines where we sequence the entire genome. We all know that Human Genome Project, where the entire genome of human has been sequenced. So the similar way the microbial genomes can be sequenced to the plant genomes, the animal genomes, the entire genome can be sequenced. In genome, you will get the information of the entire genome. You can download them, you can do analysis with them. We will not go into that detail, but now you know that how to identify a bacteria. If someone give you the name of one bacteria, you can search them. You can, in NCBI, in GenBank database, you can make their phylogenetic tree and you can have the idea with which bacterial species it is showing maximum similarity or maximum identity. Oops. Satoshi, how much time we have?
You can go through like 10 minutes more.
No, if it's like this, so probably I will go for 10 minutes more, and the rest, probably the protein part, we can do in the next class, Santoshi.
Yeah, that would be better.
Okay.
Actually, it is like a great and wonderful talk because with experimental, as you are showing, that would be like a great help for them also. So
after the presentation, I will share the PPT also in the BIXS group so that they can have it.
Okay.
So we were still here, isn't it? Till now, this part is clear. When I will share the screen with you, PPT, this is a part of results where today you did a result, isn't it? The result that you did today of the gene, of the identification, all the step that you have done, I want to see here which bacteria you have chosen, and you download the PDF, you take the screenshot and put the phylogenetic tree till here. So this I want that you all do as a homework until the next week we are going to have the class. So I'm going to, for the protein will be another week, but this you put your results here so that I can see that you all are doing it. And if you feel any problem after that also, let me know. Okay, so we till now know about genes, the nucleotide sequences, and we know that how to identify a bacteria via sequencing. Now, the microorganism also have protein. The bacteria also have protein. And how to see the proteins? Because we now know how to see their genome, how to see their DNA, but we don't know how to see their proteins. And protein is also a macromolecule we talked about, and proteins are really interesting because all the enzymes in our body, all are proteins. Proteins basically are making our daily life going on. And in case of microbial proteins, they have different uses. Imagine, in case of us, probably a hemoglobin is a protein, actin, tubulin, albumin, they are protein. In case of microorganism, they are proteins or enzymes we are using in day-to-day life. How? We are producing, probably you all know, insulin via Escherichia coli expression system. So we are using the microbial proteinary system to produce our own proteins. Also, their enzymes like catalase, amylase, protease, urease, all these enzymes are getting used in food industry, in wastewater treatment, in biomedicine, in different industry. These enzymes are getting used. Also, the good factor of why we use microbial enzymes, many times the microorganism that lives in high temperature and they have high temperature stability, we prefer to take the enzymes or the protein from those bacteria. Why? Because normally we know that proteins or enzymes are functional till 37 degrees centigrade, the temperature of our human body. Over that, the enzymes start degrading, isn't it? The enzyme lose their action, the enzyme don't function. But when a microorganism or a bacteria or archaea lives in a very high temperature of hot spring or fumarola or mud volcano or hydrothermal vent, the temperature stability of the organisms or of those organisms are high. So when the temperature stability of high, living in 120 degrees centigrade, living in 80 degrees centigrade, the enzymes of those organisms also have higher temperature stability. So now probably you all have heard this RT-PCR, isn't it? RT-PCR for COVID-19. In RT-PCR kit, there is an enzyme called Taq polymerase. This Taq polymerase is a thermally stable DNA polymerase. And what this Taq polymerase do? This Taq polymerase do the amplification of a particular gene, and in thermal cycler machine, the temperature is high, 90 degree, 80 degrees centigrade. Taq polymerase don't get degraded in that temperature. How the name Taq come? From Thermus aquaticus. This is a bacteria, Thermus aquaticus, was isolated from the hot spring of Yellowstone National Park in United States. And Thermus aquaticus lives near 90-degree temperature. So because Thermus aquaticus lives in 90-degree temperature, for their thermal stability, their enzymes are preferred in the industry for additional thermal stability. Because in industry, all the machines works in high temperature. So this is the help that the high thermally stable enzyme gives to us to be used in any industry. My cats are making a lot of sound, so if you are hearing some meow meow, please sorry for this, because when in the morning I speak, they start doing this cat sounds. Okay, so in to understand protein structure, proteins are normally in 4 different structures: primary structure, secondary structure, tertiary structure, and quaternary structure. 4 different levels of organization. What is the primary structure? Let us think about where. Probably we all know spring. In the pen, if you open, you can see this kind of secondary structure spring, isn't it? It's very common seeing this kind of spring. If you make the spring totally straight, this is the primary structure. A straight structure is primary structure. Example, insulin. Insulin is a polypeptide, and the insulin peptide, this hormone, is a primary structure of protein with 151 amino acids. So this is a simple amino acids joined one by one makes the primary structure of the protein. Simple straight structure. When this simple straight structure are getting coiled, they form alpha helix, beta strand, pi 10 helix, different type of other coils, and they are basically, they make an angle, the angles are called psi and phi angles, and these angles basically interact with one another and get stable as coils, get stable as sheets. This phi and psi angle, the secondary structure of the protein, was developed, the idea was developed by an Indian scientist called Ramachandran. And Ramachandran plot is a very famous plot to identify protein structure, to get to know about protein structure. So have been done by an Indian scientist. What is a tertiary structure? When you basically mold this spring, it becomes a tertiary structure. Probably you all have seen hair girders of plastic like this. Carl normally use this kind of hair bands of plastic, like a spring but circle, a good example of a tertiary structure, where the spring itself is molded in another structure. So all the protein in our body like actin, tubulin, albumin protein for eggs, the egg albumins, are all tertiary structure of protein. And what is a quaternary structure? You take two tertiary structure together, can be hemoglobin, can be DNA polymerase, and when you add them two together, it become a quaternary structure. So you can see that there is one secondary structure of a different color, and one primary, secondary and tertiary structure of a different color, another of different color. When you add them two together, they become quaternary structure. Think like you are playing with a wire, then you are trying to bend it, trying to make it as a spring. When you have the wire is a primary structure of protein. When you make a coil with that wire, it's a secondary structure of protein. You are playing with that wire and suddenly you are bored. You don't want to play anymore. So what you do, you crumble the secondary structure and you throw it. So that scrambled secondary structure is a tertiary structure. And two different kind of protein structures you add together becomes a quaternary structure. So these are the four different organization of a protein: primary, secondary, tertiary, and quaternary. And via bioinformatics, through the bioinformatic tools, we can know all these structures. We can see all these structure, we can see the composition, we can determine which structure it is. So in the next class, we will play with those things to see all the primary, secondary, and tertiary structure via bioinformatic tools. With probably next day, you come with the idea of any protein, each one of you, one of the protein from bacteria. So we will, if you are 3 people or 4 people, we can play next day with 4 different proteins from a microorganism. So next day, I will of course come with the idea of mine, I will show you, but then with the enzyme or the protein that you will let me know from bacteria, we will do this, the work, the practice here with yours, so that we can have bigger idea how to do with it. Okay? Okay till now?
Yes, ma'am.
Okay. So if there is any problem while doing this part of the identification, just write in the BIXS WhatsApp group, and I will shortly be back there. And I will also share the presentation in the WhatsApp group.
You can switch on your video so we can take some nice photos. Huh? Santoshi can take photos?
Yes, sir.
Yes, will be good to see them also. Otherwise, feeling like the kids, where are they? Cannot see them.
All of you switch on your video
Have any
so we
questions
can take
you can
photos.
ask right now? Or
No, please ask. Please ask. Yes.
Naya Mehta, can you switch on your Sri Sharma as a join? Hello.
So can you see me now?
Yes.
yeah,
Yeah, can take
I
photo.
can see.
Someone?
Yeah.
Santoshi?
Yes, sir.
Hey, Rajul. Rajul has come. Maybe you make a grid, bigger grid, then it is easy to take, right?
I have taken also, yes. So you can ask all. If you have any queries or question, please ask now.
Yeah, now if you make a bigger grid, you can bring everyone here. So go, Indrani, can you switch on your video? Rajul? So go, Indra. Ready?
I've taken print screens.
Renu? Sugu? How about Renu and Sugu? They don't want to show their face? Yeah. Hey, Renu and Sugu, can you show your face, please? Actually, 1 minute, I'll tell you why I need this photo. Renu and Sugu, can you switch on your video? Okay, I think they are not available. Because when I was telling about these classes, right, some bank here keen to know who are the children who are studying this course, and there is a possibility they will expand this to collaboration in other region like Singapore, Malaysia, all these places. So they want some photos, proof that how for each our semester students or kids are. Later I'll tell Santoshi and all.
Yes, sir.
We need all the photographs and things. So then we can write up to them, and they are going to post it in 18 countries actually, all these photos, and they want to show how children are going through all these things we are doing and things. They are very. Today morning we had a discussion on that. Some kids were there. 4 kids came, Bhavya, Irnadam, Nabia and Fahad. They all attended the meeting, and it was very good with the bank because for children, for your information, they can sponsor your bachelor degree here. You can do your undergraduate, and the bank is keen to give sponsorship for those children who are coming excellently, like publishing. As some of you know, we are trying to publish some papers, right, with the previous batches. So they're very surprised to see how school children can do all this. So they want to bring talent to Singapore and also make your undergraduate study sponsored by, I don't know, partially or fully. So you have a good fair chance in all this. We are trying our best to cope up with them. So we need more photographs and presentations. So it will go a few rounds. I don't know when it will be ready. By the time you all finish your +2, or then it will be a time that you'll be able to get a sponsorship and can study in Singapore or some places. Okay, so that's what I want to tell in a brief. Later we will talk internally with Aparna and Santoshi and Renuka, all this. Maybe tomorrow. Today I feel very tired. Okay, so that is good news for all of you. Some banks, they are talking in terms of 1 or 2 million. So it's big money actually. Anyway, it will take some time. It's not so easy, but yeah. So they can escalate Bioclues to other region. That's what they are talking, and they say it's going to be a good move initiative now. Okay. Okay, I don't want to delay. The lecture was so good. I already told Aparna's lecture, even I missed the initial part. So I have to rush and listen. It was really very good.
It's really amazing, sir.
Thank you.
I think my kids might have enjoyed that.
Practical thing, right, Aparna?
Yeah.
What, sir?
When is the practical for this?
Yes. I'm going to share today the presentation in the BIXS WhatsApp group. So there is a part of results where they should put their results of today's of identification and this, and the next day they will do for the proteins.
Okay.
But next day we have the entire time to play with the, probably me and Santoshi, we can both discuss about this and can think on this. But the next day we have entire time to do the practicals.
Okay, yeah, that will be good because that's where the value comes when you all do the practicals. Okay, Naya and Aradhya or all the children here. So it will be very good to do a practical thing. Then you can do a lot of things. Okay. Take care. Bye bye.
Do you have any queries?
No, ma'am.
Okay, okay.